Hello and welcome. My name is Philip Lewing, and some of you probably do know me. And if you don't, I am the scientific director of the CIFST Food Cluster. And on behalf of CIFST, Canadian Institute of Food Science and Technology, welcome to our 2025 Food Cluster Webinar Fall Webinar Series. And today, our sponsors are basically this webinar is being sponsored by Lactalis as well as BSA Weiberg. So thank you all for being here today. I know that in Toronto here, it is not snowing, so it's a nice sunny day. And so the topic is also a sunny topic. Today's topic is onion peel powder in food applications, bridging knowledge gaps for natural shelf life extension and food waste reduction. Now, we are very, very honored to have our speaker today. It's Dr. Alexandra Grigorchik. I usually mess up her last name, but I think I've been
You got it.
practicing. Thank you, Alex. Uh, Dr. Grigorchik is a research scientist in a consumer sensory and market insight at Vineland Research and Innovation Center. So, welcome, Alexandra. Please tell us a little bit about yourself before we start the presentation.
Thanks, Philip, and hi, everyone. Thanks for joining us. Um, as Philip mentioned, I'm a research scientist at Vineland Research and Innovation Center. I have a background in food science, and I've been at Vineland for about 13 years now. For the past 5 or 6 or so years, a lot of my research has focused on food waste reduction, working with growers, but also CPGs and other food companies to better understand what they can do with their byproduct streams and looking at ingredient development and also working with other teams at Vineland to look at how to use some of these byproducts for things like soil amendments or other purposes. And that's what brings us to today's presentation, which is again about repurposing food processing byproducts.
Fantastic. Now, for the audience, if you have any questions for Alexandra during her presentation, we encourage you to actively engage by posting your questions in the Q&A tab at the bottom of your screen. Now, you don't want to hear from me, so I'll pass it over to Alexandra.
Okay, I'm just going to share my screen. I hope you can see that.
Yes, we can. Yeah.
Okay, wonderful. So first a bit about Vineland before I jump into the presentation. Vineland is a Canadian organization that's dedicated to horticulture, science, and innovation. And we are an applied research organization, which means that we work very closely with industry. We tend to sit in that gap between innovation, upstream innovation like in academia and between there and industry, where we like to bring innovations that are closer to commercial stages. And so also being an applied research organization, we tend to work very closely with industry partners, either through grant-funded research with industry investment or through direct contract service research. Nearly all of our projects have an industry partner on them. And that's how we try to stay relevant to meet our mission, which is to improve the economic viability, sustainability, and competitiveness of horticulture in Canada. We're an independent not-for-profit organization, which means that we're not government, we're also not academia or industry. We sit somewhere in the middle of those three categories. In terms of research capacity, we have a 218-acre campus. And we do call it a campus because we have 35 buildings that include research labs, research farms, and greenhouses. And we have around 100 employees, of which 60 or so are in R&D. So as you can imagine, with around 60 people in R&D, we have quite a wide range of expertise at Vineland. We have researchers that focus on consumer and sensory market insights, market research, but also in sustainability and soil health, plant production, engineering and automation, as well as biochemistry. And as I mentioned earlier, part of our mission is to improve the sustainability of the horticulture sector. And we do this through a portfolio of research topics that we cover that include themes like environmental metrics, like life cycle assessments, developing soil management practices for growers that are more environmentally friendly, but also waste valorization, which is the connection to today's presentation. And within the waste valorization space, we've done some work around ingredient development, where we focus on developing cost-effective but value-added functionality from fruit and vegetable byproduct streams. We've also done quite a bit of work around opportunity scoping, so working with either byproduct generators like processors or growers to help them understand what the possible potential is of those byproduct streams and ways to better use it. We're also working with ingredient companies that are looking to produce an upcycled ingredient, but don't really know where to get the material from or what kind of byproduct would be the best fit for their needs. And so within that opportunity scoping, we also work in a connector role, connecting different parts of the supply chain to each other for new partnerships. And we've also done sector overviews where we identify different sources of byproducts and report on those more publicly to generate more awareness. And we've also done studies to better understand the industry-wide challenges preventing the growth of, or that can support the growth of the upcycled food space. So last year, Second Harvest put out this really wonderful report that looked at the different sources of food waste within the Canadian food supply. And people tend to assume that most of the food waste occurs in consumers' homes. But in reality, that actually only accounts for about a quarter of the food waste that occurs through the supply chain. And about 2/3 actually comes from the food industry, and it happens before even reaching retail. And so the food industry actually has a large role to play when it comes to being able to address food waste. And within Canada, nearly a third of this pre-retail fruit and vegetable waste is deemed unavoidable. And so unavoidable means that it's this byproduct that's generated inherently due to processing, like peels, pumice, or grade outs, so fruits or vegetables that don't meet the specifications that are required for processing or for the grocery retail, fresh market. And so they could be too small or could have aesthetic imperfections or so on. And what makes these byproducts very interesting is that they generate recurring and predictable volumes of byproducts, which is of interest if you're trying to make a new food ingredient, because you have a reliable source of raw material. And there's tons of research, published research out there that has looked at some of these byproduct streams and it found that they're rich in antioxidants and hydrocolloids or antimicrobial, some of them. And so it's pretty well documented that many of these byproduct streams have valuable components in them. However, there is also still a knowledge gap preventing commercial valorization because many of these haven't actually become commercial products yet. And onion byproduct is an example of this. In Ontario alone, there's a couple major onion processors that together generate over 5,000 metric tons of onion peeled byproduct annually. And from what they've told me, they expect this volume to increase in the coming years just due to the growing demand for processed onion. But what's interesting is that onion is known to have antimicrobial properties, and that's not a new finding. I remember learning about this in my undergrad, almost two decades ago. And so, most of us probably have heard of it, of onion having antimicrobial properties if you're in the food industry. And in our literature review through this project, we found there are over 30 scientific papers that are published that discussed the antimicrobial properties of onion. And they looked at a variety of formats. They were looking at some were on onion peel, some were on onion flesh, some looked at onion extracts or onion homogenate. And it was, quite a wide range of papers. Some of them looked at these antimicrobial properties in, a petri dish, but some also took it to the next step. Because, of course, we know that sometimes when you see antimicrobial properties in a petri dish, they don't always translate to a real product. But there were 11 articles that did examine microbial suppression of onion in food formulations. And what's especially interesting is that 10 of these 11 found positive effects. There's quite a bit of consensus that onion does have these antimicrobial properties, even when it's put into a food formulation, not just in a petri dish. But despite all of this evidence, to our knowledge, there's no onion-based antimicrobial available on the market. And in looking at the literature, we did identify some unknowns and knowledge gaps that could be preventing this byproduct from becoming a commercial product. For one thing, we couldn't find any literature that discussed the sensory differences between onion powder, like the traditional common onion powder, versus onion peel powder. Because a lot of the research was done on onion peel powders or onion peel extracts, because the peel contains more antioxidants than the flesh. But there's no good understanding of how the sensory properties change if you're consuming peel versus the onion flesh itself. There's also no research on the impact of drying method, whether you're looking at different dryer types or drying temperatures, and how that impacts sensory or antimicrobial performance. Another gap we identified was in understanding what the performance is from an antimicrobial perspective in at realistic concentrations. So, for example, there are lots of studies that have looked at, as I mentioned, 11 studies looked at antimicrobial properties in a food formulation, and some of them have inoculated those samples with, say, E. coli or salmonella to see if the onion peel powder can kill these pathogens. However, there's no data to support that the concentration they were using is actually realistic, and if it's even palatable. So, for example, there was one study that looked at salmonella kill rates in burgers where they added onion peel powder, but the onion peel powder was added at concentrations of either 8.5 or 12%. And I don't know about you, but I don't think I've ever cooked a meal where I had 12% seasoning in my dish. And so that doesn't seem particularly realistic. And so I think there's a need to dig into that a little bit further. And so that brought us to this current project, which was a collaboration with Gum Products International. They're a Canadian company that's based out of Newmarket, Ontario. And as their name implies, they produce various gum blends. And they also produce a line of clean label antimicrobials. And they were a really great company to work with because they have a bake lab, they have a meat lab, and they also have a level 2 micro lab where they could do some of this testing. And so they were interested in testing onion peel byproduct as an alternative clean label antimicrobial. For today's presentation, I'm going to do a bit of a deep dive into onion antimicrobial research that we did and share those results. And I'm going to very quickly highlight some additional research we did on onion peel powder functionality. So that texture research was actually on a wide variety of different crops, but we did find that onion peel was one of the best performing ones when it came to this work that we were doing. So I want to also highlight that, as that could be another secondary benefit of using onion peel powder as a ingredient. So starting with the antimicrobials research, we did this research in 3 stages. In the first stage, we examined the impact of processing conditions on onion peel powder sensory properties. And we used that research to inform our selection of processing conditions moving forward. Next, we use those processing conditions to determine a maximum, to prepare some onion powder, and then determine the maximum palatable concentration in pork breakfast sausages. And that involves some consumer testing to figure out at what point, how much can you add before it becomes less palatable. And then we use that concentration to inform the last stage of our research, which was the microbial challenge testing, where we looked at the effect of onion peel powder on spoilage and how it affects the natural growth of organisms that are already present in the sausage. And then we also looked at what happens if you inoculate the sausages with salmonella or with E. coli. Starting with this first phase, we obtained onion peeled byproduct from a local commercial onion processor, which we received frozen. And then we prepared it into powder by using 2 temperatures, a lower temperature and a higher temperature, and also 2 dryer types. Then we used these powders in rapid sensory profiling, and we evaluated the sensory properties both in when we dispersed the onion peel powder in water as well as in a vegetable broth. So here's a sample of our findings. This was research that we did in the vegetable broth. We added the 4 versions of onion peel powder that were dried using different methods. We also had a commercial onion powder for comparison. And so you can see this onion powder shows up twice. That's because we used it 2 times because all the samples were presented to our panelists blind. And so by putting the onion powder in there twice, we can get a sense of validity of our results. And because the 2 onion powders are showing up close together on the sensory map, that means our results are valid. We also had a blank with nothing added to it, just the vegetable broth and also onion flesh that was dried. And so the way you interpret this plot is similar to a PCA, if anybody is familiar with that, where these data points, the closer together they are, the more similar they are to each other in terms of sensory properties. And the further apart they are, the more different they are in sensory properties. And this plot on the right shows a projection of how different attributes relate to different areas on the map. And so you have to, in your mind, imagine overlaying these 2 figures. And so what we found was that onion peel powder version 1 was much more similar to traditional onion powder, and it had high onion aroma, high garlic aroma, whereas these 3 other versions of onion peel powder that were dried differently had more astringent perception, bitterness, more green type aromas like celery and pepper. And so we did find from this work that the different drying conditions led to different sensory properties, with some becoming closer to commercial onion powder, whereas others were higher in bitterness and vegetal notes. So based on that, we chose that one drying method that was produced attributes closer to traditional onion powder, and we used that in the next stage of research. And so in this stage of research, we wanted to find out how much onion peel powder can you add to a product before negatively impacts consumer preference. And we chose to do the work in pork breakfast sausages. And we chose that product pretty intentionally because we know that in pork breakfast sausages, it's a pretty mild flavor. I know pork is not a very strong tasting meat, and pork breakfast sausages don't have a ton of seasoning in them. Whereas something like, I know a kielbasa, you could probably add a lot more seasoning to it and mask the onion powder in it. A pork breakfast sausage is a lot less forgiving. And so it was a bit of like a worst case scenario where we can't add a lot of seasoning. We wanted to know what's the minimum amount of onion powder that you can, I guess, maximum amount of onion powder you can use in that kind of context. Because of course, the more onion powder you can add, or the more of an antimicrobial you can add in a food system, the more likely you are to see antimicrobial properties and to have it be effective. So you want to maximize that, but you also don't want to detract from consumer liking. So GPI prepared this pork breakfast sausage formulation for us, and we supplied them with the onion peel powder, which they included in the pork breakfast sausages at various concentrations. So we had 6 concentrations ranging from 0% to 7.8% of onion peel powder in the sausages. And so next we did a consumer rejection threshold test, which is where you present consumers with various pairs of product. In this case, we chose our control to be the 0% formulation, and we presented increasing amounts of onion peel powder in the breakfast sausages. We had 25 participants, and the products were evaluated in the sensory lab under red lighting. So if there is any effect on color from the added onion, it was masked by the red lighting. And each pair was presented in sequence, and each time consumers were asked, which sample do you prefer? And so presumably, if you ask the percentage of onion peel powder increases, initially you might see a positive reaction from consumers because you're adding a little more onion powder, a little more seasoning, a little more flavor. But eventually it gets to an inflection point where it's just too much. And so it starts to negatively impact their preference. And so that's the inflection point that we were interested in finding. And what we found was that the inflection point was 1.43%. So it's not super high. So when you think back to some of the work we found in the literature, where they were testing 8.5 or 12%, that was clearly not very realistic. And in fact, a lot of the research that we found was testing at around 3 to 5% of onion powder or onion extract. So whereas in reality, we're finding in a pork breakfast sausage, the threshold for palatability was 1.43%. So we then took that concentration and applied it to antimicrobial challenge tests. And here we had, so GPI produced those pork breakfast sausages again. This time we had a control sample with no added onion powder. We had a sample with commercial onion powder at 1.43%. And then we had two other sausage formulations that each had 1.43% of an onion peel powder that's produced either using drying method 1 or drying method 2. And so just a note that before starting the work, we did measure the water activity and pH of the samples, and they were all the same. So the effects that we're seeing is not due to differences in water activity or pH. And so next we put those, GPI put those breakfast sausages raw into a refrigerator. And every so often they measured the growth of various organisms to see what effect the onion peel powder was having. And so here are the results for the total plate count, which looks at the overall population of bacteria in the sausages. On the y-axis, we have log CFU per gram, so the concentration of bacteria. And on the x-axis, we have time, so the day of sampling. And this, again, was in raw sausage, because that's how we envision that if you're producing sausages and you sell them to a store, they will sit in the refrigerator at the grocery store or in consumers' homes raw until they're consumed once after cooking. And so what we found was that once you start looking at later on in the storage time, say day 14, day 21, you had the onion peel drying method 2 powder was actually quite effective in reducing the concentration of bacterial load in the samples. And it actually induced almost a 1 log reduction in microbial population. And so based on this, it seems like the onion peel powder could actually be used as a shelf life extender to prevent spoilage or slow down spoilage. Next, we looked at pathogen suppression. So the first thing to note is that when looking at the growth of pathogens that were inoculated into the raw sausages, even the control sample reduces in pathogen concentrations over time, because over time you have growth of spoilage organisms that produce lactic acid, so it's lactic acid bacteria that produces lactic acid. And so if the acid is produced in the sausage, that can kill off some of the pathogen. And so what really we're looking for here is how the test samples compare to the control, and do they reduce the growth of Salmonella or E. coli faster than the control sample? And unfortunately here we didn't see any differences, any major differences. You can see that they pretty much stay very similar all throughout. And so we did not see any major effects on killing off of Salmonella E. coli from the onion peel powders. However, what is interesting though is, if you remember from the previous slide, the onion peel powder had lower concentrations of spoilage organisms. So despite the lower concentration of spoilage organisms, which means there's less competition for the pathogens, we're still seeing a similar behavior in terms of E. coli concentrations in these samples. So that's also noteworthy, that even though it doesn't kill off E. coli or Salmonella faster, it also doesn't let them grow any faster than the control sample would. So overall, we found that the threshold for consumer rejection of breakfast sausages containing increasing amounts of onion peel powder was relatively low at 1.43%. But despite this low concentration threshold, it was still effective against the growth, effective in reducing the growth of spoilage organisms, and so it could be used as a shelf life extender. However, it had no major effect on pathogen growth. Next, we're jumping into the texture research. So this research initially focused on this enzyme called transglutaminase. I'm going to just call it TGAs for short. And TGAses are ubiquitous in nature. They're found in plants, in animals. Microbes produce TGAses. They're in the human body as well. But what's relevant to the food industry is that microbial transglutaminases, TGAses, are used commercially, typically derived from microbial fermentation or cattle blood fractionation. And if you ever, it's a protein cross-linking enzyme. If you've ever heard the term meat glue, that's what it refers to. Sometimes it's called meat glue, but the enzyme behind that is TGase. And as a protein cross-linking enzyme, it's used for binding and gelling. So, for example, it's sometimes used in the deli industry, so making deli meats to stick different cuts together. It's also used to make surimi, which is that imitation crab meat that you often find in sushi, like in California rolls. It's also used in the plant-based food industry to make plant-based scrambled eggs or plant-based hot dogs. It can also be used in the dairy sector to either increase the thickness of low-fat yogurt and prevent syneresis in yogurt. And it can also be used in bread production for dough stabilization during proofing, so the bread dough doesn't collapse. And what draws to this to look at this enzyme is that back in 2006 there was a paper published that looked at apple pomace and its content of TGAs. And there's a lot of papers out there that have looked at various plant sources and how much TGAs they contain. But this paper was interesting because it went a little bit further beyond just looking at does it contain TGAs, yes or no, and how much. It also looked at it freeze-dried apple pumice powder, into a powder and then added this freeze-dried pumice into a pork sausage. And they found that when they measured instrumental firmness of this sausage, it was higher than in a control sample. And so they suggested that there was some texture effect of this freeze-dried apple pumice, and that it could be from this enzyme. And this caught our attention because using a dried powder as a TGase alternative would have several advantages. First of all, dried powder production is less costly than existing commercial TGase alternatives because they are produced through fermentation. So that requires fermentation, isolation, and then powder production. Additionally, if you're using microbial transcontaminase, in many jurisdictions, including Canada and the US, it has to be included on the label. And so writing transcontaminase on the label doesn't sound great to consumers. Whereas if you're using a dried apple powder, you could just put apple powder on the label, which sounds much better. So for our research, we decided to validate the research findings, but we wanted to go a little bit further than apple pumice, and we screened 12 different fruit or vegetable byproducts for TGase activity. And then we did some work to understand the conditions for activity and temperature tolerance. So for example, looking at how much TGase activity you can detect at different temperatures or with or without calcium addition. Next, we picked the ones that had the best performance at that stage and moved them forward to examine enzyme stability. So we took 4 different crop types, byproducts, and we dried them into powders using commercially relevant drying methods, so not freeze drying. In this case, we used either a fluidized bed dryer, a vacuum oven, or convection drying, and used 3 temperatures for each of those to see how the activity, if the activity was retained and how much after those different drying approaches. And then finally, the best performing ones out of those tests were moved forward to stage 3, which was to validate their functionality in a plant-based food formulation. And so for today, I'm focusing on just stage 3 in the interest of time. And so here, one of those top performers was onion peel powder, which is why I'm talking about it today. And so I'm only going to be focusing on the onion peel powder results in this presentation. And so what we did to validate the texture effects is we added various fruit or vegetable powders, including onion peel, into plant-based patties. And they were added at a concentration of 1.75%. We also had some control samples where we added either 1.75% maltodextrin or MTGase, just for the basis of comparison. Next, we did sensory testing, where we did a difference from control test. And in this case, panelists were presented with 2 samples at a time. First, a control sample, which was our microbial transcontaminase sample, along with 1 test sample. The test sample could either be a blind control, which was our MTGase, and this was done just to validate our sensory results, where we had panelists unknowingly evaluate MTGase versus MTGase. Other test samples included the maltodextrin sample or the samples that had fruit or vegetable powders in them. And then panelists, for each pair of samples, they were asked to rate your perceived difference from the control. And then next, if they found a difference, they were asked to indicate any difference in intensity of the following attributes. And so when they were evaluating these attributes, we found that both adhesive and cohesive had significant differences. And so those are the attributes I'm going to be focusing on next. So here are the results for cohesive and adhesive. And just to note that cohesive was defined as the degree to which the mass holds together, as opposed to breaking, crumbling, or cracking. And so you can think of it, a cohesiveness as the resistance to crumbling. So for example, something that's pasty and stretchy would be very cohesive, whereas something like a meat patty, it tends to break apart more. So that would be less cohesive. And adhesive was how sticky it was, how much it sticks to your mouth surfaces and teeth. And so we found that the maltodextrin samples, for example, were a lot more pasty and kind of gummy. And so the way you interpret this information in this plot is the closer to zero it is, the more similar it is to the control sample, which is our MTGA sample. And so as we had hoped, the blind MTGA sample compared to itself was virtually zero. So that means that our sensory data is valid. And the maltodextrin sample was significantly different from the MTGAs, as you can see by the asterisks. Whereas our onion peel powder samples were not significantly different from the MTGA's controls. So this told us that based on our sensory testing, the onion peel byproduct powders produced plant-based patties that had cohesiveness and adhesiveness that were similar to patties that contained microbial TGAs. And to further support this research, we also did some testing with using a texture analyzer, a cut test. And from the cut test, we saw that the mean firmness for onion peel powder was again not significantly different from the MTGA's control sample, but it was significantly different and higher than the maltodextrin sample. So the key takeaways from this section were that the onion peel powder showed promise as a texturizer in plant-based patties, and the texture effect that we observed was comparable to MTGA's control samples. Of course, based on this research, we can't actually confirm if the texture effects were directly from native TGAs in the powders, but we were selecting samples that had higher levels of this enzyme activity. And so in terms of potential future research directions, we'd love to be able to better understand the contribution of native TGAs to texture modification versus other components that are potentially in that onion peel powder, but also to test the effect in additional formulations and to see if we can repeat this effect in other contexts. So overall, what we found is that onion peel powder is a promising food ingredient. There's large volumes of onion peel byproduct available in Ontario, and there's ample evidence in the literature that it has antimicrobial properties. Through our research, we found that dried onion peel powder had a meaningful effect on suppressing spoilage organisms, even when we used it at a realistic concentration in pork sausages. And so it could potentially be used as a shelf life extender. And then finally, we found that it also, in addition to having these antimicrobial properties, it also has a texture effect, and it can modify texture in plant-based patties. So I'd like to acknowledge everyone who contributed to this work. At Vineland, we had, I know, quite a team that contributed to it. We had Amy Blake and Amy Jenkins, Christine, Jean, Craig, and Sarah. And then from Gun Products International, we had Thomas and Henry working with us on this project. And of course, I also want to acknowledge the funders, the Food Science Cluster through the Sustainable CAP program and OMAFA. Thank you.
Thank you very much, Alex. Appreciate this wonderful topic here in terms of onion peel. I have a few questions. There are a few questions on the Q&A. Let's start with those first. This one person asked, do you have any data regarding the reduction of lactic bacteria?
Unfortunately, we didn't look specifically at lactic acid bacteria. We just looked at the total plate count in general, but that could be something to look at in the next phase of the research.
Okay, thanks. Now, with respect to the onion peel, does the age of the onions, could that make a difference in terms of the functionality? I know this might be a tough question here. It might be a stumper.
Well, it's not something we've looked at, but we do know that in general, if you're storing, so in general, we know that the antimicrobial properties in these types of products tend to come from the antioxidants in them,
Hmm.
and antioxidants do degrade over time. So,
Right.
it's reasonable to think that if the onion peel is older,
Yes.
you might have less antimicrobial properties.
Right.
However, the onion peel, so onion is available most of the year because it's harvested in the fall, but they store it for around 10 months of the year for processing.
Mm. There's another question. Someone asked, what level of TPC will be considered spoiled?
So, so I'm not a microbiologist, but from what I've been told from GPI, they
Okay.
consider anything with a TPC of 6 to be six or higher to be spoiled. And so we actually did find that only the sample that contained the onion peel byproduct with drying method too, uh, stayed below that threshold of 6, even after 21 days.
Okay. I'm not trying to stump you here again, but um, analysis for TGA's activity in, let's say, in the onion, was that conducted? Did you have a method in terms of how that was determined?
Yeah, so we did that using an assay. This assay is in the literature. We pulled the assay from the literature and then just modified it to perform better in a plant context because
Okay.
most of the assays for TGs are developed for animal systems.
Right.
That's to do with a bit of modification, make sure it's working appropriately in a plant system. But there was an assay, and we plan to publish the results in the next few months. So that methodology will be available.
Okay. There's another question, and probably this ties in with one of my question too. It from the data that you've shown in terms of one of the drying procedure seemed to be more effective in terms of the performance here. So someone mentioned that the onion peel is an industrial byproduct. What is a practice that is recommended for making it safe to consume?
Yeah, we so it is a fresh product, so it's fresh processed. And so typically, um, when this onion is processed, it's processed and consumed raw. So, like for example, some of these processors that are processing the onion, they chop it and then make chopped onion flesh.
Right.
Uh, a diced onion that you can buy in grocery stores that's frozen. And in terms of the peel itself, that's also another question that we'd have to look at. But with the drying methods, we did use drying methods that our lowest temperature that we used was, I trying to recall, around 55 degrees Celsius, but it goes on for a couple hours. And so you have to, you could then look at, the killer rate for any potential pathogens in that product after that drying time. It's definitely a question that still needs to be investigated a little further, just to confirm that the product is safe. But generally, with powders, it's lower risk.
Okay, I've got another question. So I'm sure there are lots of other value-added components in the onion peel. I know you probably haven't tested them, but are you able to speculate in terms of what other value-added components are there that could be looked at for future?
Yeah, sure. So onion peel surprisingly actually contains quite a bit of pectin. I know apple hummus is kind of famous for containing pectin, but onion peel actually has similar or maybe a little bit more pectin in it than apple hummus from what I saw in the literature. However, for pectin to work, you do need, of course, acid and high sugar, which of course we didn't have in a plant-based patty. So we don't think it's the pectin that's causing this effect. There's also, I think it was another food cluster research project actually that was looking at how polyphenols in grape can interact with proteins to impact texture. So it's a
Hmm.
possibility that maybe the antioxidants are also interacting with proteins to create, some texture effects. I'm just speculating here. Something that would have to be looked into. But definitely there's, more than just the antimicrobial properties that are, potentially of interest in this onion peel byproduct.
Hmm. Um, you're probably not going to like me. So 1.43% was you found as the threshold in terms of where sensory says this was the threshold here. Have you tried going above that and probably masking some of um, the unacceptable flavors to see if it really helps in terms of microbial reduction here or in terms of the pathogens? I know you stopped at 1.43 because of sensory says above that it tastes bad. Oh,
Yeah.
yeah.
Yeah. I'd love to do some work to figure that out because it's very, of course, pork breakfast sausages, as I mentioned, are pretty mild tasting, so you can't add a
Yes.
lot to them. But, there's also potential to use onion peel powder and things like more seasoned sausages, which
Hmm.
could probably add a lot more to them. And so it's reasonable that, maybe in other products, maybe you could see effects on pathogens. It's still an open question.
Sure. Absolutely. I've been asking a lot of the questions here. Anyone else in the audience there would like to jump in? Um, give you an opportunity. Here's your chance to ask all the questions you've been thinking about in terms of onion peel powder to Alexandra. Okay, if there are no other questions, thank you very much, Alexandra. Appreciate it. And on behalf of CFSC, we thank you for your informative presentation. Yes. Our next food cluster webinar is on Wednesday, December the 3rd at noon. And the topic is fermentation of mushroom waste to produce umami extract. And the speaker will be um, Zainab Razavi, and she's affiliated with Food Development Group as well as the University of Guelph. Now, registration is now open on our website, and of course, it's free to all CIFST members. Now, to all the audience there, we appreciate your active participation today and looking forward to have you again in the upcoming session. So thank you, Alexandra, for giving us this very informative. I believe that onions, how you presented not only in terms of the ability to affect texture, but other functionality in terms of microbial reduction here. This is very, very interesting here. So thank you again on behalf of CFSD. Enjoy the rest of the day, and thank you very much for attending. Cheers. Bye-bye.